All right, this video is going to be about lipid absorption, goes along with the digestive system. And we just talked about carbohydrate absorption, just talked about protein absorption. The thing about those two is they get absorbed directly into the blood. Lipids do not do that. Lipids are absorbed into the lymphatic system first. So a lipid is a fat, we know. And fats, of course, are hydrophobic. Hydrophobic. So when we eat fat and we take the fat into our digestive tract, which is really watery, lots of water in there, it will separate just like your salad dressing would separate. That's oil and vinegar. Okay. So it will separate out and it will form these big old fat globules. So this over here is a fat globule. So all the fat just accumulates together and forms these big old globules. Well, to chemically digest lipids, we have a enzyme that is secreted by the pancreas, and that is called lipase. And lipase is actually hydrophilic, okay? So lipase, number one, needs help in order to work on and digest the fat. And number two, it has to have some surface area to do that. So this big old fat globules, all the fat is inside of it. So if the lipase were going to try to digest this big old fat globule, it would only be able to contact the outside here. It wouldn't get anything inside, okay? So that's where we have something called bile that comes into play. So bile is a substance that emulsifies fats. And basically emulsifying the fat makes it so that it breaks up that big old fat globule into lots of little particles, okay? So bile is made, it's got cholesterol in it, it's got phospholipids in it, it's got other stuff in it, but the main important thing about emulsification is it has bile salts in it, which are cholic acids. So these bile salts are what break up this big old fat globule into these smaller little droplets here. So if we add bile to this fat globule, it breaks it up into these little droplets here. And this is where the lipase then can contact the fat, these smaller things, so that it can digest the whole fat instead of just the outside of that big globule. So again, the lipase is hydrophilic, so it does need a little bit of help. So lipase has a helper molecule called colipase or colipase, and it will contact the little droplets, and that will allow lipase to be able to contact the droplet as well. So once lipase gets on these fat droplets, it breaks it down. It breaks these down into free fatty acids and monoglycerides. And it forms with these things called micelles. And these are little tiny molecules of phospholipids. This is a layer of phospholipids, and they surround the fatty acid or the monoglyceride. And they're condensed, so they're really, really tiny. Once we get this micelle here, the micelle then can enter the enterocyte because it's a fat, and fats can go through the plasma membrane without any help. So it can go into the enterocyte, which is the small intestine cell, and when it gets in there, it goes into the smooth endoplasmic reticulum. Smooth ER. And if you remember about the smooth endoplasmic reticulum, it is a organelle that processes fats, makes fats, cholesterol, and things like that. So once these micelles are inside the enterocyte, they go into the smooth muscle, the smooth ER, and the smooth ER then recreates the triglycerides, which are fats. So it reassembles the triglyceride. And if you remember, a triglyceride is a glycerol backbone, so a glycerol with 3 fatty acids attached to it. Get rid of that. Okay. So 3 fatty acids attached to it. That is a triglyceride. Once it forms these triglycerides, then it puts them in this thing, and this is within the enterocyte. It places them inside a sphere of phospholipids, because it's got phospholipids. So it puts them inside here. And it also sticks in there, it's a good color for this, puts in there cholesterol. So cholesterol and triglycerides are in here. Okay. And then it contacts the rough ER and gets some proteins. And it takes those proteins and places them just like this along the phospholipid layer here. These are called apoproteins. So apoprotein. And again, we've got the apoprotein, we've got cholesterol, and we've got our triglycerides. And this entire thing, this whole crazy looking sphere of phospholipids and proteins and all of that, this is called a chylomicron. And essentially this chylomicron is a big fat lipoprotein. It's a lipid and a protein together. And this thing, this chylomicron, which is, we're inside the cell, so we're inside the cell in here, and the chylomicron will go into the lymphatic system in this structure that's within the digestive tract called a lacteal. Once in the lacteal, then it goes up to the thoracic duct or whatever, and then gets dumped into the veins, the subclavian vein, whatever it is, and then it goes into the blood system. Okay, once it's in the bloodstream, this chylomicron thing gets to the liver, and in the liver capillaries there are receptors, and these apoproteins link to receptors on the endothelium, which is the epithelium of a vessel. So the endothelium has these receptors and it links onto that endothelium. Those endothelial cells have a enzyme called lipoprotein lipase. So they contain lipoprotein lipase. And that lipoprotein lipase then breaks down this stuff again into free fatty acids and glycerol. It breaks down those triglycerides. This is it within the liver. The liver then can decide, okay, well, we need some energy. I'll just send those fatty acids over to the muscles or whatever so that they can get some energy, because we can get lots of ATP from a fatty acid. If there isn't a need for that, or a need for some type of, something for fatty acids, a use for it, then it will repackage it again, and it will repackage it into triglycerides, and it will then send it to adipocytes for storage. So if it does that, it will use, again, lipoproteins. Okay, so lipoproteins basically carry lipids around the body. There are basically 5 different types, but the main 2 that we are familiar with are the LDL and the HDL. So the LDL is low density lipoprotein, and the density means the amount of the concentration of the fat inside of the lipoprotein. So if the concentration is high, it is a low density lipoprotein. If the concentration is low, we're talking about a high density lipoprotein. The low density lipoproteins carry fats from the liver to fat cells, to adipocytes. HDL lipoproteins carry lipids or fats from other tissues back to the liver to be recycled and excreted. Okay, so that's why it's good to have a lot of high density and not so much of the low density lipoproteins. Low density lipoproteins are important. You just don't want too many of them because they do bind to those receptors on the inside of endothelium, and if there's too many of them, it can cause plaquing, which is cardiovascular disease. So once it gets into the adipocyte can store it and we can use it for later. So we can tap into our fat stores when needed. So this is lipid absorption. It's a little more complicated than the carbohydrate and the protein absorption. And just keep in mind that it does not go straight into the blood. It goes into the lymphatic system first through those lacteals that are specific to the digestive system.